Today we're going to be looking at Chapter 18, Part A. This chapter is still over the cardiovascular system, but now we're going to specifically be looking at blood vessels. So let's introduce some terminology. First and foremost, blood vessels are a delivery system. They're dynamic structures, and we're going to look at that structure and function relationship that they have. But we want to establish first that they begin and end at the heart. Arteries are actually blood vessels that carry blood away from the heart. Veins are blood vessels that carry blood toward the heart, and capillaries are going to be at the intersect between arteries and veins. Capillaries are small networks of blood vessels that are in direct contact with tissue cells, so they're able to directly serve cellular needs. Arteries always carry blood away from the heart, and a way to remember this is that arteries start with A, so arteries, A, away. Veins always carry blood toward the heart. We see the word in the term veins. So that's a way to remember that veins are going to carry blood back toward the heart, where blood will enter or go in to the heart. Now, statements that cannot be all statements include the oxygenation of the blood in arteries and veins. We can say usually or generally or for the most part when it comes to the oxygenation of blood vessels. For example, almost all arteries carry oxygenated blood, except for the pulmonary arteries and the umbilical arteries of the fetus. Similar types of things can be said for the veins. Veins almost always carry deoxygenated blood, except for when it comes to the pulmonary veins and the umbilical veins of the fetus. So we can make some all statements, but be aware of the ones that don't follow through with the way that the cardiovascular system works. Here we can see the lymphatic system's relationship to blood vessels. We have, for example, cardiovascular capillaries, but also lymphatic capillaries that are closely associated. The lymphatic capillaries are actually able to help the cardiovascular system retrieve some of its lost fluid. And we'll talk more about the lymphatic system in Chapter 19. So let's move on though and look at the structure of blood vessels. And we'll start with the structure of blood vessel walls. All blood vessels contain a lumen, an open space where you would find whole blood. So that's just the blood containing space. And it's surrounded by a wall. However, this wall is not singular. It's generally a system or group of walls that surrounds the lumen. So the walls of all vessels except capillaries have actually 3 layers. They're much more complicated than a simple kind of boundary between whole blood and outside space. The 3 tunics of most vessels are the tunica intima, tunica media, and tunica externa. Capillaries only have a tunica intima, or 1 layer of endothelium, and it may have a sparse basal lamina. And if you can't remember what an endothelium is, it's a layer of simple squamous epithelium located in areas such as the inside of the heart and the inside of blood vessels. So let's look at each tunic in more detail. The tunica intima is the closest to whole blood, or you could say it's the most intimate with whole blood. It's the innermost layer of a blood vessel wall. This innermost layer is made up of endothelium, again, simple squamous epithelium that lines the lumen of all blood vessels, even our capillaries. It's continuous with the endothelial cells of the endocardium, and that would be inside the heart. And the fact that it's continuous with the endocardium is in order to reduce the generation of friction. So we have a very slick surface that reduces friction as we go from the inside of the heart chambers to blood vessels. That way we're not introducing massive amounts of friction or peripheral resistance in the cardiovascular system from the beginning. This tunica intima also contains a subendothelial layer. This is kind of a supportive connective tissue basement membrane, but we really only find a subendothelial layer in large blood vessels. So vessels larger than 1 millimeter. Next, we're going to talk about the tunica media. So think media, muscle, both start with M. That's a good association. Be aware that blood vessels contain smooth muscle, not skeletal or cardiac muscle. That's often a fact kind of misintroduced or kind of misstudied, I find, when students are looking at chapter 18, because they're coming off of chapter 17, which is all about the cardiac myocytes or the comparison of cardiac to skeletal myocytes. And so it's important to just make sure that you know that blood vessel walls contain smooth muscle, not cardiac, not skeletal. The tunica media is a middle layer. It's composed mostly of those smooth muscle myocytes, but also, especially in the larger blood vessels, sheets of elastin. The sympathetic vasomotor nerve fibers that go to blood vessels are able to innervate this layer. So sympathetic is of the sympathetic nervous system. Vasomotor is, think of it as vessel movement. Vaso is a common prefix for blood vessels. And what the sympathetic vasomotor nerve fibers are able to do are control vasoconstriction and vasodilation. So two terms that you should know the definition of. If a blood vessel constricts, it's going to decrease its lumen diameter. So vasoconstriction, it just means blood vessel constriction. Vasodilation is blood vessel dilation, where we see an increase in lumen diameter. Now, usually the tunica media is the bulkiest layer, and because it can constrict and dilate, it's responsible for maintaining blood pressure and blood flow. And last but not least, the outermost layer of a blood vessel wall is the tunica externa. So tunica externa, think externa like exit or external, but it's also called the tunica adventitia. You may see both terms. And mostly the tunica externa is for protection, for reinforcement, for providing an anchor to surrounding structures. It's composed mostly of loose collagen fibers, and it is infiltrated with nerve fibers. Similar nerve fibers to that of the tunica media, but we find a looser association here, and there may be some lymphatic vessels that kind of innervate or weave through the tunica externa. A lot of large veins also contain some elastic fibers in the tunica externa, but those are not always found and are not always found in arteries as counterpoint. The vasa vasorum is a vessel of the vessel structure. So what that means is that really large blood vessels are so large that they're not able to get nutrients in their outermost layer from diffusion. And why would they use diffusion to get nutrients? Well, there's blood running through these blood vessels, and it's pretty close to most of the layers, but in large blood vessels, the tunica externa is so far away from the blood of the lumen that diffusion cannot take place in order to provide nutrients. So there's actually even tinier blood vessels in the tunica externa of these large vessels, and they can be called the vasa vasorum because they're a system of tiny blood vessels found in larger vessels that function to nourish the outermost external layer. Here in figure 18.2a, we see the generalized structure of arteries, veins, and capillaries. The artery is on the left, the vein is on the right, and not labeled in this image is a capillary, which is what we see down here, kind of between the two, so medial and inferior. Some general characteristics to note when you're looking at a histograph of an artery versus vein, for example, is that an artery is going to look much more structured. It's going to be more round, maybe what you expect from a histograph of a blood vessel, whereas a vein is going to have a collapsed appearance. You can also see this dark purple line or ring that goes all the way around the artery. This is the tunica media. So the tunica media is much more pronounced, it's bigger, it's thicker in arteries than we see in veins. And on kind of a counter note, we would say that the tunica media in veins is minute. It's not really there. There is muscle, but a lot of veins are not responsible for changing blood pressure or blood volume, specifically through blood flow, by constricting or dilating. We can see a lumen in both, the open space where the blood would be, and you can see that to an extent there's more tunica externa in the vein than there is in the artery. That's not as obvious as with the tunica media here in purple, but tunica externa is more pronounced or abundant in veins as opposed to arteries. All right, we're going to go ahead and move forward. And we're going to look at kind of the more clip art version of the structure of arteries, veins, and capillaries that your textbook gives you, which is figure 18.2b. So we can see there's a little bit more to arteries than veins. We have a thicker tunica media in arteries as opposed to veins. We have more elastin in arteries as opposed to veins. The structure we haven't talked about but will are valves that we find in veins, not in arteries. We also see the vasa vasorum, or small blood vessels, in the tunica externas of both arteries and veins. If we positioned the heart up here at the top, we would say that blood flows away from the heart and the arteries to smaller and smaller arteries that connect to capillaries, and then blood flows out of the capillaries into veins. It flows into bigger and bigger veins on its way back to the heart. So that blood is going to enter the heart from the vein. We can also just pay quick attention. Here we have one layer of endothelial cells, or simple squamous epithelium, supported by a small layer of basement membrane or connective tissue. Next, we're going to be looking at different types of arteries based on their function. For example, we have elastic arteries, muscular arteries, and arterioles. Elastic arteries tend to be the largest as opposed to muscular arteries and arterioles. Elastic arteries also are going to have more elastic tissue than these other types of blood vessels seen in this image. The muscular arteries are going to have more smooth muscle as opposed to the elastic arteries and the arterioles, but the arterioles are going to be mainly responsible for constricting or dilating in order to control blood flow and blood pressure. And we'll see why that is in a couple of upcoming slides. Eventually, we'll also talk about different types of veins, which are a little more simpler information there than compared to the arteries. And let's also look at the capillaries. So the capillaries are just endothelium. There may be some supportive connective tissue. Venules are small veins. When two venules merge, or even multiple venules merge, we get an actual vein. And so the larger veins have more smooth muscle than, say, a venule. And we're definitely not going to see much elastic tissue in venules, but we see a little bit in veins. We can also see that the vein has a lot of fibrous tissue or collagenous tissue that's generally found in the tunica externa. This belies the fact that we have a larger or more abundant tunica externa in veins than we see in arteries. So let's look in more detail at the elastic and muscular arteries as well as the arterioles. So these are the three groups that we divide arteries into, and we're basically dividing them into these structural groups based on their size and their function. So elastic arteries are the ones closest to the heart. So we can include the aorta and its major branches. Elastic arteries are definitely thick-walled. They have large low-resistance lumens, and they are also called conducting arteries because first and foremost, they conduct blood away from the heart. They are going to conduct blood away from the heart to medium-sized vessels, the ones we're probably going to look at in a second, called muscular arteries. And for elastic arteries, we find elastin in all three of the tunics, hence their name. So elastic arteries are able to expand and then recoil because of the elastin found in all three of their wall layers. We see most of this elastin in the tunica media, and it contains substantial smooth muscle in the tunica media, but is relatively inactive in vasoconstriction, even though its tunica media with most elastin is going to have a lot of smooth muscle. It's mostly there to expand and recoil. Elastic arteries expand and recoil, and that means they are great as pressure reservoirs. That expansion, that recoil, is going to make sure that as blood is ejected from the heart, it is then pushed to further branches of the arterial system. And that makes sure that continuous blood flow is possible. Blood flow is going to go downstream to kind of smaller arteries and eventually to capillary beds. Next, let's look at the muscular arteries. So elastic arteries are going to give rise to muscular arteries. So downstream from elastic arteries are these muscular arteries. So even though there is quite a lot of tunica media in our elastic arteries, we find even more in comparison in our muscular arteries. And that's because they're delivering blood to body organs. And that's going to be more tunica media compared to their size. Now, the only reason that elastic arteries have more tunica media overall is because they're so big. The diameters of a muscular artery range from pinky finger size to pencil lead size, and they account for most of the named arteries. So things like the brachial artery or the dorsal pedis artery, those are going to generally be muscular arteries. They have the thickest tunica media with more smooth muscle, but less elastic tissue. So these are not elastic arteries, but we do have that association between them since elastic arteries give rise to muscular arteries. The tunica media is going to be sandwiched between elastic membranes. So the muscular arteries can be active in vasoconstriction, but they're not the most important arterial structure in vasoconstricting. Arterioles are the smallest of all arteries. They're going to have larger and smaller sizes because we have large arterioles that contain all three tunics and smaller arterioles that are mostly a single layer of smooth muscle surrounded by some endothelial cells. They do control blood flow into capillary beds, and that's why their vasoconstriction or dilation is most important, because constriction of an arteriole can restrict access to a capillary bed, or vasodilation can allow access of blood to flow into a capillary bed. So this control of flow in capillary beds via vasodilation and vasoconstriction of smooth muscle is very important for the regulation of microcirculation, which is blood flow through a capillary bed. We would also call arterioles resistance arteries because of their changing diameters. They're essentially changing resistance to blood flow. And first and foremost, it's important to know the structure or location of arterioles, and then it's important to know their function. So arterioles lead to capillary beds. We will see then the blood flow into a capillary bed, and that will be kind of our intersect, or we'll call it anastomoses, between arteries and veins. So next we'll look at capillaries, which makes sense because we've just left the arterial system. Capillaries is what our arteriole allows blood to flow into if it's vasodilated. And capillaries are very small. They're microscopic. Their diameters are so small that sometimes red blood cells have to kind of squeeze through, and they would use that plasma membrane protein called spectrin in order to pass one at a time. The walls of capillaries are just a thin tunica intima or endothelium. In the smallest vessels, we might see one or two pericytes, which are spider-shaped stem cells, stabilizing the capillary walls, but fewer and far between depending on the size of the capillary. The pericytes are also able to play a role in vessel repair. And another interesting thing about capillaries is they supply almost every cell, because cells, which make up tissues, need nutrients. They need things like glucose and oxygen, among other things, in order to carry out cellular respiration and create ATP. And ATP is important for cellular metabolism. However, we are not going to see capillaries in cartilage, certain types of epithelia, cornea, and the lens of the eye. Now, I said certain types of epithelia because oftentimes once you study epithelium, you know that we're still going to have a certain amount of vascularization up to that epithelium. So the epithelia is not completely cut off from nutrients. It's able to get from connective tissue vascularization nutrients via diffusion. The function of capillaries is very simple: gas exchange, or exchange of some type. Now, I said gas exchange because that's going to be very important later on. I kind of want to just put it in your brain that gas exchange is one of the most important things that capillaries do. But exchange in general is the function of capillaries. So the functions include exchange of gases, nutrients, waste, and hormones, which we talked about recently in chapter 16. And this would be exchanged between the blood and interstitial fluid. And then from the interstitial fluid, which bathes cells and tissues, we would have those nutrients either go into cells or leave cells. There's different types of capillaries, And even though they all have a similar structure, they're made up of endothelial cells, which are joined by tight junctions. They generally have some gaps between their endothelial cells called intercellular clefts, and all of these things allow for passage of fluids and small solutes. There's even more structural modifications depending on a specific type of capillary you're looking at. So the types of capillaries are continuous capillaries, fenestrated capillaries, and sinusoid or sinusoidal capillaries. Continuous capillaries are the most abundant in the skin, your muscles, your lungs, the central nervous system. They're continuous capillaries because we don't have really hardly any intercellular clefts, or we could say the gaps between endothelial cells are smaller compared to the other two types of capillaries. We also know that continuous capillaries of the brain are unique. They are almost completely enclosed. They have tight junctions between their endothelial cells and no intercellular clefts, not just small ones. And this is actually because continuous capillaries must form or work with astrocytes to form the blood-brain barrier. So continuous capillaries are the least permeable and most common. This doesn't mean they're not permeable. It just means that depending on their location, they're more or less permeable. For example, in the central nervous system, they're less permeable than normal, but they're slightly permeable in the skin and muscles and the lungs. They generally have stabilizing parasites. They may have penocytic vesicles to ferry fluid across the endothelial cells. So that's a type of active transport that most cells can perform. And the term continuous means they have the least breaks. So they're the most continuous in structure. They're going to have smaller intercellular clefts. They're not going to have any pores, which are called fenestrations. And their structure is important for certain areas like the central nervous system. And our example given for that was the blood-brain barrier. So I'm harping on that because a common question is, what type of capillary helps to build the blood-brain barrier? And that answer would be continuous capillaries. Next, we'll look at the fenestrated capillaries. These are a little more leaky, so they're more permeable than continuous capillaries. They're found in any area that is involved in active filtration. Great example there would be the kidneys or any area that's involved in absorption, like the small intestines. Glands that secrete hormones generally are going to have fenestrated capillaries, and these endothelial cells contain fenestrations if they're a part of fenestrated capillary. That's where they're getting their name. So endothelial cells that contain pores or fenestrations compose fenestrated capillaries. And these pores are not intercellular clefts, so that means that a fenestrated capillary contains intercellular clefts between endothelial cells, and the endothelial cells themselves kind of look like a piece of Swiss cheese and contain these pores or holes. And the fenestrations allow for increased permeability. And we do find that fenestrations are usually covered with a thin kind of glycoprotein diaphragm, meaning that they're slightly protected. They're not just open holes for things to leak out. So fenestrated capillaries have holes or fenestrations. They occur in areas of active filtration and absorption. Fenestrations are pores or tunnels. Even though they're slightly protected and contain a glycoprotein diaphragm, they are essentially ways for things to get out other than through penocytic vesicles and really small intercellular clefts. We do see that oftentimes the intercellular clefts of fenestrated capillaries are slightly larger, but not necessarily larger than that of continuous capillaries. And then last but not least, we'll look at the sinusoidal capillaries. There are definitely fewer tight junctions when we're looking at sinusoidal capillaries. So the endothelial cells themselves are held less tightly together. We definitely have fenestrations, and these pores are much larger than we saw in the fenestrated capillaries or the continuous capillaries, which we had none in the continuous capillaries, some in the fenestrated. And the intercellular clefts are broad. We even have incomplete basement membranes, and so larger lumens, larger fenestrations, larger intercellular clefts, few tight junctions. All of this coming together and building a capillary bed means that we have a very permeable structure. We have a structure that is going to allow blood flow through it, but that blood flow is going to be sluggish. But there's a reason for this. We find sinusoidal capillaries in areas like the liver, the bone marrow, spleen, the adrenal medulla. And these are areas where we want to allow time for modification of large molecules or structures that can pass between blood and tissue. This modification may mean that phagocytosis by macrophages occurs in order to destroy any type of pathogen that may have been absorbed. So oftentimes sinusoidal capillaries are not only the leakiest of the capillaries, they also contain macrophages in their lining to capture and destroy foreign invaders. So here we have in figure 18.3c, a sinusoidal capillary only found in certain locations, liver, bone marrow, spleen, and adrenal medulla. They have large fenestrations, or large pores, large intercellular clefts, or spaces between endothelial cells, and very few tight junctions, which is a special cell junction that's impermeable, holding their endothelial cells together. So we would see that their irregular shape and large lumens makes them very permeable, but also allows blood to only move through slowly. So kind of to summarize before we move forward, a capillary bed is an interwoven network of blood vessels between arterioles and venules. So a capillary bed does consist of capillaries, and it's going to be at the intersect between arterioles, which are small arteries, and venules, which are small veins. Microcirculation is a term you should know. It's the flow of blood through a capillary bed from the arteriole to venule. Microcirculation is not necessarily talking about exchange, just the flow of blood through the capillary bed. A terminal arteriole is the arteriole that branches off and leads to a capillary bed. And a post-capillary venule is what blood from a capillary bed drains into as it enters the vein or a venous system. Between a terminal arteriole and a post-capillary venule is where you find the capillary bed, and a capillary bed may have 10 to 20 capillaries or exchange vessels that form its entirety. And flow through a capillary bed is controlled by vasoconstriction or vasodilation of an arteriole. So it's controlled by the diameter of that last arteriole that leads to a capillary bed called the terminal arteriole. The local and chemical conditions at the terminal arteriole and the innervation via the sympathetic nervous system to the arteriole are going to be able to regulate the amount of blood entering the capillary bed. So if the arterioles above and at the terminal arteriole are dilated, then blood is going to be able to flow into the capillary bed, and this occurs when blood is needed by a tissue. We would see that an arteriole upstream and the terminal arteriole are constricted if we need to shunt blood away from a capillary bed because the tissue does not need blood. So here's the general anatomy of a typical capillary bed. We can see, for example, we have an arteriole here that's dilated, which allows blood to flow into the capillary bed, give away its oxygen, so that's why it's going from red to blue. And then as the blood flows out of the capillary bed, it's entering a post-capillary venule, and then that will move into larger vein structures, like a even bigger venule. So arteriole, upstream arteriole, terminal arteriole, capillary bed, post-capillary bed, venule. And we have blood flow or microcirculation through the capillary bed when the terminal arteriole is dilated. When the upstream and terminal arteriole are constricted, there's no blood flow through the capillary bed, and we will have subverted or not had blood flow through this capillary bed into a tissue. There is another type, or we'll say another structural component to certain capillary beds, and those are for the capillary beds found in serous membranes, specifically of the intestinal mesenteries. And we won't really talk about the intestinal mesenteries until we get to the digestive system chapter. But we want to acknowledge the vascular shunt system and the precapillary sphincters that work together to route blood through the intestinal mesenteries of serous membranes. So here we can see an upstream arteriole, the arteriole that leads to the capillary bed, so the terminal arteriole, and how it's possible if we close these muscle sphincters or constrict them, that in this special type of capillary bed found in the mesenteries of the digestive system, we could get blood to flow through the capillary bed, but it would flow through the shortest route possible. And that would be using the vascular shunt. So to use a vascular shunt system, we would have to be looking at capillary beds located in the mesenteries of the abdominal cavity wall. We would have to be able to see precapillary sphincters along the vascular shunt, and we would have to have vasodilation of the terminal arteriole, but constriction of the precapillary sphincters. So what's happening here is this tissue does not need any oxygen, or maybe it doesn't need nutrients, but whatever it doesn't need, we're shunting blood straight to the venule. We're going to get it there as fast as possible. But we're going to shunt blood to the venule by actually having the blood just flow through the capillary bed as quickly as possible, but not into the kind of outer parts of the capillary bed. These outer parts of this capillary bed are called true capillaries. And if the blood flow isn't getting there, we're going to have little, if any, actual exchange. So here we have the upstream and terminal arterioles dilated. Blood flows into the vascular shunt system, which includes a meta-arteriole and a thoroughfare channel, into the postcapillary venule, and then a larger venule. The reason that the blood flows straight through the vascular shunt system and not to the true capillaries is because these precapillary sphincters are closed. Let's go back 2 slides. A precapillary sphincter is smooth muscle surrounding access to true capillaries. True capillaries branch off a meta-arteriole of these capillary beds located in the membranes of intestinal mesenteries. We're going to control this via local chemical concentrations, so it's not going to be controlled by the nervous system. And again, it only occurs in certain circumstances, but just be familiar with these terms. Don't let them catch you off guard. You should know what a terminal arteriole is, precapillary sphincter, meta-arteriole, thoroughfare channel, postcapillary venule, true capillaries, and also that the meta-arteriole and the thoroughfare channel are 2 parts of the vascular shunt system. All right, so let's move away from capillaries now and talk about veins. Veins, we know, carry blood toward the heart. Blood's going to come back to the heart, enter the heart, go in to those heart chambers. And the formation of veins begins after the capillary bed, when the capillaries unite at the postcapillary venule. Then the postcapillary venule merges into a larger venule, and when many venules merge, that's going to create veins. Venules are smaller than veins. Again, they're connecting the capillary bed to either larger venules or veins themselves. Venules are going to be very thin. They're not going to consist of much more than endothelium, but they're probably, depending on the size, there's probably going to be a little bit of smooth muscle and probably some pericytes to help stabilize a postcapillary venule. Postcapillary venules especially are very porous, so they do allow fluids and white blood cells into tissues. Whereas the larger venules, they're going to have more pronounced tunics or layers to their walls, and we definitely say that they have smooth muscle cells. So veins are formed when venules converge. They have all 3 tunics, but thinner walls and larger lumens compared to arteries of about the same size. Their tunica media is thin, but we see a bigger tunica media in arteries, but their tunica externa is abundant or thick as opposed to arteries. We do find a lot of collagen fibers and elastic networks in both their media and externa. The large lumen of veins and their thin walls may give them kind of a collapsed appearance if you're looking at a histograph, but they have very large lumens, and that's great for storage of blood. So sometimes veins are called capacitance vessels because at any one point in time, they may contain up to 65% of the blood supply. So let's just get a reminder as to what our veins and arteries look like. We all see our capillaries down there at the bottom. The arteries have a thick tunica media. They have a round lumen, whereas veins have a collapsed lumen but a large lumen, a thin tunica media, and an abundant tunica externa. Then our capillaries down there, kind of the middle bottom, they have one layer of tunica intima. They may have some supportive pericytes, but other than that, they have only one tunic. If we look at the relative proportion of blood volume throughout the cardiovascular system, we can see in this image, figure 18.6, why veins or the venous system would be called kind of a blood reservoir, or that the blood vessels that make up the venous system are considered capacitance vessels, just because at any one point in time, these venous blood vessels contain a large proportion of the blood. So let's look at the structural component of veins. They're generally going to have a lower blood pressure, much lower than arteries. They don't have as thick a tunica media. They don't play a large role in vasoconstriction or vasodilation, and their large diameter lumens offer little resistance to blood flow. Other structural adaptations that veins have are they have valves. The valves we find in veins are called venous valves, and they prevent backflow of blood. So that backflow of blood would be attempted because of gravity, and venous valves are going to prevent that. We find, therefore, most venous valves in the veins of limbs. Venous sinuses are flat and kind of pocket-like structures of veins. They generally have extremely thin walls. One such venous sinus that you've come into contact with or seen before is the coronary sinus of the heart. We'll also eventually talk about the dural sinuses of the brain. Venous sinuses are thin, so they're kind of similar to capillaries in that they are only composed of endothelium. Make sure that you review, after looking at arteries, capillaries, and veins, this structure, and you understand why the arteries versus veins have different structural components. And also make sure to pay attention to capillaries as an arterial venous anastomose or junction between arteries and veins. Table 18.1 shows you the three types of arteries we looked at. Elastic arteries near the heart, which lead to muscular arteries, which are many of our named arteries, and that leads us to our arterioles, which are our smallest arteries, and they are involved in the vasoconstriction and dilation that actually impacts blood pressure and blood flow. So make sure to pay attention to arterioles, especially the terminal arteriole that leads to capillary beds. Pay attention to the differences between venules and veins. The postcapillary venule would drain a capillary bed, and when many venules converge, we would generate a vein. A clinical homeostatic imbalance we want to mention is just varicose veins. These are dilated, painful veins, usually due to something wrong with the venous valves. They can become incompetent or leaky. There's different factors that contribute or may lead to someone developing varicose veins. There's some hereditary factors and other just kind of lifestyle factors. But they generally are slightly hereditary, and they're conditions that can hinder venous return. But things that may be environmental are just prolonged standing in one position, or obesity or pregnancy. But this generally occurs because blood pools in lower limbs, it weakens valves, and we get a dilated or painful vein because of the leaky affected valves. This affects more than 15% of adults. Elevated venous pressure can also cause varicose veins. A reason that someone may have experienced elevated venous pressure include straining to deliver a baby or having a bowel movement that raises intra-abdominal pressure. So any type of straining can cause varicosities in veins that either result in varicose veins, or if this occurs in anal veins, can produce hemorrhoids. Some terminology we want to establish just before we finish Part A of Chapter 18 is the term anastomoses. The term anastomoses means interconnection of blood vessels. So a vascular anastomoses is an interconnection of either different types of blood vessels, maybe elastic to muscular, or we can have, for example, anastomoses between arteries and veins. An arterial anastomoses is in order to provide alternative pathways for blood flow to tissues. There's fewer arterial anastomoses than there are venous anastomoses. Veins like to form junctures much more than arteries. We may also call these arterial anastomoses collateral channels because they would be used when another artery is blocked, but they should lead to the same location or tissue. We find collateral channels most common in joints, abdominal organs, the brain, heart, but none in the retina, kidneys, and spleen. An arteriovenous anastomosis is essentially a capillary bed. It's what's going through a capillary bed, especially what we saw in the digestive mesenteries. So an arteriovenous anastomosis is the shunt in capillaries, that vascular shunt. So it's, for example, the meta-arterial, an artery structure, to the thoroughfare channel, which is a venous structure. And then venous anastomoses are so abundant that pretty much if any vein gets blocked, it's okay because there's going to be another channel that allows blood flow back to the heart. So rarely is a clot or an occluded or blocked venous anastomosis or a venous vein structure going to be a problem because of all of the venous anastomoses that do exist. And that's it for chapter 18, part A. It is mainly anatomy, but make sure to familiarize yourself with arteries, capillaries, and veins before moving on to part B.